This is a soil microscopy exploration webinar. It's a kind of a mini workshop where we'll be looking at samples, all sorts of different soil microscopy samples from compost and soils, and we'll be exploring some of the soils that we've been building and some of the things that we found that are really show dysbiosis too. So you're going to get the best of worlds of healthy soils and really nasty soils. We'll be exploring the biology that lives within them. So this is going to be a really good look into what lies inside of our biology and why this is important. And you'll get kind of an understanding of why we're looking at this and the impacts they can actually have on your growing and your composting and how to improve some of those things. And just a quick intro before we get started today. This is for Compost Academy, and we have a great program coming up. Oh, Seth, good to see you, man. Great program coming up called the Microscopy program. We start on December 11th. It's a 6-week live training program with also self-paced microscopy there. I'll drop the link and we'll do a little promo at the end. But if anyone wants to join during this webinar, you get $100 off. And if you're a returning student from Compost Academy, you get $150 off because we love having this community kind of expand and grow and help each other out. So I'm just popping that link in the chat and then we'll get started here. Okay. Yeah, so that link there should give you $100 off. Brian, you can only chat with the hosts and panelists. You should be able to chat with everybody right now. Let me know if you can't.
Yeah, I can't. I only see
Oh,
host
weird.
panelists and just the host or panelists in there right now.
Hmm,
Yeah,
okay. Well,
but I
while
can
you're talking,
see the chat coming in.
I'll
So
try to fix that for you.
thanks, man.
Fix it on my end. Well, why don't I hand it over? Keisha or Brian, do you guys want to start giving an intro about the
Sure.
microscopy process?
I'll start since I'm the one who has the non-functioning microscope program today. I'll get my words in early. I was planning on sharing with you guys all the samples. We just got back from building 6 windrows in Southern California with some students of Compost Academy. Actually, they're starting a new compost lot. And we've been exploring their eucalyptus-based compost, which is something, we always teach don't use eucalyptus. We showed up on site. We had 500 yards of eucalyptus chips only that we had to work with. And there was actually an ecosystem after a month. So I wanted to show you guys what was in it. It's actually really unique. But I think I'll end up sharing it on Instagram instead. So you guys can follow us at compost.academy or Catalyst Bioamendments and just see the microscope updates I'll put up in the next week. Um, but yeah, I'm Keisha Ernst. I work with microbes all the time. So why is the microscope important to me? It validates everything I do. I've got a compost company called Catalyst Bioamendments, where we basically grow microbes for farmers and people that restore land. We work with Brian Vaugh. We have a consulting company where we do really all kinds of interesting, super large scale projects. And then, we teach about it as well. I've got a lab, and so there's all the technical side of it. I have to use it for my work. But for the purpose of this class, I actually love it for exploring. And I think I've like been an explorer spirit since I was born. I mean, my first memories are just like looking through the bushes, like climbing the trees, making up crazy stories about like what this land used to be. Like I've always kind of been that character. And, I think I got into probably like middle school, high school, and I became aware that all the exploring was kind of done. Our teachers were like, all right, the new exploration, the new frontier is tech. And I was like, tech? Boo, I don't want to work in computers. I was bummed for like a good 10 years. I thought, what am I going to do for a job? I want to do something exciting. I want to share my adventure. And thank goodness for Dr. Elaine Ingham. She helped me learn how to use a microscope and set my passion ablaze. And, I would say we've been working in this industry for a long time now, and we're having a lot of success. And I boil it all down to the microscope, but really, the reason we keep going, because we weren't successful forever. Anyone in this industry knows when you first get started, it's not like being a plumber, it's not like being a carpenter or working in tech. There's no direct line to a job. It's not like, oh, on this side of it, I'm going to be fixing someone's toilet for $85 an hour. It's very much something you have to build, and it can be frustrating, it can be overwhelming. It's hard. But you know what it is? It's fun. As long as you have the microscope and you have the spirit anywhere similar to mine, if you like to look around and see new things and things you've never seen before, things you don't understand, yeah, the microscope just keeps it fresh. So for me, it's been a tool to keep me going, and then it's also been my tool to make money. So to me, the microbes are everything. I'm a little bit obsessed. To be honest, I'm trying to back my personality away from it a little bit at this point because I'm like full-blown microbes. But yeah, I don't know. I'm excited to share with you guys. It's so cool. There's 51 people here today. Very neat to see this many microbe farmers showing up for just a live event where we look at soil. So thanks for being here. And I think I'll pass it off to Brian.
Sweet. Thanks, Keisha. And you know what? It's so funny because it's like Tale of Two Cities. My childhood was different, and our personalities are definitely very different. I am very data tech driven. So I actually had a career in the high tech industry, and I'm all about analysis and analyzing things to the nth degree and comparative type of stuff. But the beauty of it is you can use a microscope in both those kind of spaces. And to be honest with you, Keisha is really rubbed off on me in the fact that I've become much more adventurous in I'm now a much more curious person. So thank you, Keisha. That was very well. And a little bit about my history with the microscope, which was kind of interesting. My wife and I, we had a homestead down in Northern California, and we were managing it very conventionally, and I wasn't happy with it. So we decided to go to regenerative practices, starting really with permaculture, and we were seeing differences and changes. But for one thing, it was really bugging me is I could see the changes, but it really wasn't driving to why these changes were occurring. That's when I found about Dr. Lane Ingham, started taking courses from her, and she reintroduced me to the microscope because I took some microscope classes or used the microscope when I was in college. But other than that, I had not used it in any kind of practical application. And I started looking at soils and I started comparing, different soils on my property of garden beds that were doing good or areas that weren't doing so well. And I could see differences between the two. And that my analytical part of my brain started really firing, going, okay, I can really now start to quantify some of these different differences between this different soils that I'm seeing. And that really just kind of clued me into the microscope is such a powerful tool. And it's continued on. I mean, in our work that we do, as consultants, I use a microscope all the time. I'm getting soil samples from my customers, and we are doing trials and tests, and we're doing comparative analysis, and the microscope is an essential tool. And then here at my house, I use it as well. In fact, I'll show you some samples today where you can see quite significantly a garden bed that has had a lot of attention with a microbial focus versus a highly disturbed soil that has not had that kind of focus. And the microscope shows you it really paints a picture or a story. So I'm excited to be able to kind of share that skill and the knowledge that we've gained over time by looking at different soils and seeing how they behave. It is really a fantastic, fascinating tool to be able to use. All right, I think I'm going to pass this over now. Dan, do you want to introduce yourself?
Yeah, get off mute there. Okay.
Sure.
Um, yeah, I'm grateful for the opportunity to share a sample here today. I've been a student of the soil food web for a while now. I did the foundational and the certified lab technician. I'm still in the process of the consultant program, but I also last year did the Costa Rica trip with the Soil Academy here and signed up for the academy classes last year and signed up for the microscope class. I'm super excited for this process, this journey that I've been on. Um, so I kind of like Brian, I'm in the technical world. I'm a data engineer right now and looking to shift some of that skill set into the skill set with the microscopes, compost, and all the things that I've learned at Compost Academy. My wife, Janelle, worked with like Zach and Raleigh and the water stories, got certified through that. And she was like, you need to do the soil side, I'll do this side. And so, we'd love to combine all that and start to get into, we've been doing some consulting, some webinar or training, compost trainings locally here. And so just kind of getting into that, but definitely interested in the compost, the microscopes, the things I've learned. So super excited.
Right on, Dan. And yeah, I would love to. You've been able to like replicate, have this recipe you've replicated and it's worked again and again. That's so cool seeing that you've been able to do this. Like, I found my recipe. I've got
Yeah.
success. I've met regs. And I'm actually jealous. I'm like, dang it, I haven't got there yet. So that's
Yeah.
so cool seeing how far and fast you've come. That's epic. All right. How about Maria? Let's hear about you a little bit.
Yeah, I am from Chile. It's the end of South America. We are the last city, Punta Arenas, that's where I live, the last city in the south cone of South America. And I discovered Soul Food Web, and it was like the missing link that I needed to understand holistic management. Because we were working with arrieros, which are the mountain cowboys in Chile, and we didn't understand why we needed to have domestic animals in a place where there were no wild animals anymore because they killed them all. And that's when I found Soil Food Web, and I understood that all the microbes that the animals were giving to the land, they were benefiting the land. And so I started to use the microscope and see how the soils are working in different places in Chile. And we also are regenerating a vineyard in central Chile, close to Santiago, where we have one of the best Sauvignon Blanc. And we are doing a quite good job there, and we are breaking ground right now. But I am living right now in Punta Arenas, which is very close to Antarctica. And we are a port to go to Antarctica, actually. And I am starting a compost with native bark from linga, which is a tree that only is here. And I found very beautiful things that I would like to share.
We're excited to see your samples. This is going to be really fun. All right. Well, we're going to start off with Brian today, and then we're going to go to Dan and Marie. So let's see what Brian has planned.
All right, so let me go and share this out, Raleigh. Let me make sure this is coming across. And can you guys see it?
It's loading. Nice. Yeah, looks good. Why don't you just click X on
Yeah,
the
I'll go.
camera, and then we should be
Yep.
full screen
How's
there.
that? Does that come
Um,
across full screen?
not yet. Not on my end.
Let me stop sharing here real quick. Maybe when I go to, let me try this. I'm gonna try one more thing here. And now I'm gonna share it. How's that? Is that looking full screen?
Hey, there we go. Now it's perfect. Nice.
All right. So, you guys, I'm going to share with you kind of two different soil samples. One that we're looking at right now is coming from one of my garden beds. So, give a little bit of context about this because it's always helpful when we are talking about soil samples to give everybody some context behind it so that you kind of know what's been done and what is the condition of the soil. So, this is in one of my raised beds, and this is a raised bed that was created about 3 years ago. When I moved into this particular house, the soil around the house was in really bad shape. I mean, pretty much it was all early successional weedy species. There was no plantings of any sort, and it was a very high sandy soil with very, very low organic matter. And so over the last 3 years, I've been working on this raised bed by adding mulches, compost, compost extracts, and really trying to improve the soil. The other soil sample I'm going to show you is from highly disturbed soil that comes from an area where I just put in a brand new greenhouse. And this is will reflect the actual soil that I started with when I made this garden bed here. So we're going to take a look at the garden bed soil. And also another thing that's really helpful when we're talking about microscopes is to give everybody what magnification are we looking at right now. So I right now am using my 4x objective and I have 10x eyepieces. And so really what that means is I have a total magnification of 40. And you're going to see as I move through the objectives, we'll get a higher level of magnification. So I'll show you a difference between a 40 total magnification versus a 400 total magnification. But I use the 40 total magnification to scan to see if I can find anything interesting. And so when I start scanning through the slide here, I'm looking to say, hey, is there something I want to be able to take a look at? And hey, I see something on my screen. And I'm sure you guys might be able to start to see it. See that little wiggling thing right there? That's a nematode. And so I'm going to go ahead and zoom on in, and we're going to go find that nematode. So right now I'm at a 400x total magnification. And this little guy is moving all over the place. Is it coming across all right for you guys?
Yeah, Brian, your soil's looking excellent. That test amoeba was beautiful. This aggregation, look at all your humics. Super proud of you being on the coast. That's
Yeah,
impressive.
and that's here's a good point that Keisha pulled out. Look at the aggregate that we're looking at right now. And so, aggregation is a really important aspect that microbes provide to you in soil. They build soil structure. So bacteria creates all these biological glues that are going to start to piece together the sand, silts, and clays or organic matter into these microaggregates. And then fungi will weave that all together into macro aggregates. And in fact, when you look at an aggregate, you can see all the nice rich colors that are here. But look when I zoom in to this aggregate, and I think you guys can see my cursor,
Mhm.
right?
Yeah.
So what is that hiding in there? Anybody? Anybody? It's a
Looks like
fungal
some sort of
hyphae.
fungi.
And so a couple tricks you can do is you can, brighten the light source so you can try to see through that aggregate. And it's really important when you're using a microscope as well to go through the entire focal plane. So the way I like to describe it is think of my hand as the microscope slide and my other hand as the cover slip. You have a volume of liquid that contains that sample. That's that drop that I put on the slide. Well, it's not just completely flat. There's a column there. And so we use the fine focus knob to move through that focal plane so you can see different things. You notice as I go through that focal plane, I can see different things. In fact, there's a little flagellate, I think, moving around, and there it goes. It just moved right into the aggregate, right into here. So it's really important when you use the microscope, not just to be static or just look at one plane focus. You want to make sure that when you get to a field of view, that you move that focus up and down in through that sample because you can see a lot of things just by going through the focal plane that you could have easily missed. All right. So as Keisha was saying, there's some life in the soil here. We just saw a nematode, we got testate amoebas, we got fungal hyphae, you saw a flagellate earlier on the screen. So overall, I'm pretty happy with the way that this soil is actually coming about. Now, I want to throw up another sample though. And this is of soils from my property, but this is a highly disturbed area, an area that I have not done any kind of work to. And in fact, I disturbed it even more when I put in my greenhouse. And I'll show you what soil looks like when it has very little or very limited life in the soil. Let me turn that down.
It's the same
Okay, so I
across
went
both of
back
them as
to,
your microplastics.
oh yeah, no doubt. There's a lot of trash in my yard for sure when I
You can
got it.
see it.
That'd be fun. Yeah,
So
you think it's fungi, it's just a microplastic. Dang it.
oh yeah, some of the wild colors, you'll get these really bright blues or these just really wild colors. You're like, that's very artificial. But so here's an example. I went to my 4X. You can see some things, but nothing interesting really showed up. And I'm going to go ahead and go to my 400X total magnification, and you can really start to see the difference in this soil. This soil is a lot of minerals with very little aggregation. There's some small aggregates, but look how tiny they are, these little micro aggregates. And what we're really kind of missing in this case is the fungi. There's very little fungi when I look through the sample. Also, the other life, the protozoa and nematodes and so forth, are really missing from this highly disturbed soil. So this is what, a microscope can really help confirm for us. One of the things I have done, through this property and really kind of restoring it is using the microscope to kind of track that progress. As I've been putting in compost and compost extracts and looking at the soil, I'm noticing those changes. That aggregation is becoming more developed. I'm getting more of the microbial community, that soil food web is actually building into those beds. So it's really important. And in the work that I do, when I go to apply a compost or compost extract, I'll take a look at it just to make sure that it is at that microbial quality that I'm looking for. And if it's not, then I got to figure out why. Did I just mistreat my compost? Did I not, keep it watered? Did I leave it exposed? Was there some kind of contamination that was applied to the soil? So, hopefully, this it gives you kind of a picture of how you could use the actual tool itself, the microscope, to give you a lot of really good information.
Hey, Brian, could we for a second? I know you've got another sample, and there was a comment here about JMS
Sure.
or Jadam microbial solution. And it's fun. This has been a conversation over the past few days with a few different people, actually. But the idea of bio like diversity when we're looking underneath the microscope, and what preps are we using and what are we seeing? And I thought it was really interesting. And we just saw this really quickly, the first sample you put on and then this sample. So in this sample, we've observed mineral, we've observed a little bit of organic material. The color is kind of lacking. We see bacteria very clearly, and we see some microplastics that are pretty heavy. And that's what we're seeing right away. So this is really a lack of diversity. Now, your other sample, I mean, in the few times you moved it, I saw a giant testate amoeba, I saw aggregation, lots of colors, many fungal hyphae, even some clear and colorless hyphae. I actually saw that flagellate come in, and then I giggled because it just left. It was like, no, thank you.
No,
It
I'm out.
just went the other
Whoa,
way.
wait, camera
Right, so
time.
with barely moving around, and these are both aerobic samples, we can see really clearly that there was a lot more diversity in that first sample than this second one, right? But then when we're comparing something like JMS to, say, a compost tea or a compost extract that is aerobic, I think there can be some just differences to point out, and that being that, and I think maybe you could put up your anaerobic sample right now. I'm hoping that it's as
Sure.
busy as I'm assuming it is, because the
Yeah.
anaerobic samples, they often, I've played around with anaerobic. There's so many different preps, and you really do see a huge diversity of bacteria. And without looking at, say, DNA analysis, I would assume that a lot of these anaerobic ferments have great bacterial diversity and also ciliate diversity. And, there's all kinds of organisms in there. But are they aerobic organisms? Because when I look at really good compost extract, it's quite boring. There's not a lot going on. It's not like things are moving around. And, testate amoeba can just be slow. Fungi doesn't really move.
Here we go. Okay, so context on this one, and so you can see this is I haven't showed it to you yet, but I have vermicompost, so I have worm bins that I use, and we did a microscope a session 2 weeks or 3 weeks ago, and I left that dilution sitting in the test tube, just sitting there for 2 to 3 weeks, and it definitely went anaerobic. Um, Although what's really wild, Keisha,
There's aerobes
is that
everywhere.
just the hour that I let it sit since I made up the prep, it added a lot of aerobicness back into here. So a lot of the anaerobic bacteria, I was seeing a tremendous amount of spirochetes all
Oh.
over. I mean, probably 50 in a field of view type of thing. It was a crazy amount of them.
Why'd you look first? You shouldn't have opened the bottle.
I know exactly. I was like, oh.
You broke the
So
experiment with your
Brian,
oxygen.
Brian, that's a rotifer slightly out of focus there, right?
Yeah.
that is a rotifer, and you can
Yeah.
see when that rotifer is just pulling in that vortex. Rotifers are just fascinating creatures to look in the microscope. You can see that soup. It's just using those rotors to make a vortex, and it just sucks in bacteria. and you can see that it has this device that's just crushing bacteria as it pulls into his esophagus.
What kind of role and indicator does a rotifer play, like when you see it in your soil?
Oh my gosh, they're great. They're a great cleaner of the ecosystem. Look at the
Yeah.
bacteria that they're processing right now. So like as an actual job, rotifers get bred in almost every city to take care of the bacterial overblooms in our waste systems. So they're actually city workers, like they work all over the world, like they're employed. And then when you're seeing it in a sample, it's definitely indicating that there is an anaerobic environment, but at the same time, isn't it also showing us that there's predators in the system? There's things eating the bacteria. So I like to think about aerobic and anaerobic environments. They dwell right next to each other. They're not good and evil. They're the same ecosystem under different influence. So like Brian said, he took the oxygen away from this ecosystem for a long time. And you can see that it's starting to change, but the second he put oxygen in, spirochetes go down and it starts to balance out again. So there's this beautiful balance in nature that's happening all the time between these two zones. So I always see these as a good sign. If I've got some rotifers in place, I'm not super nervous that my worm, I saw somebody, Danielle says, hey, I find rotifers in my vermicompost. Like, of course, vermicompost it's super moist. There's little anaerobic pockets, and these guys will function at, eating spirilla, spirochetes, all kinds of bacteria. So I see them as just powerhouse predators, like I would, similar to the way that I would see ciliates.
It's
There's
almost like
a flagellate that's getting almost pulled into the rotifer.
I can't see. Oh, I see it.
You you see him right there?
Yeah.
He's like trying to escape. He's like, I'm getting out of here. But yeah, rotifers are just fascinating creatures to be able to observe. I mean, I have spent probably close to an hour sometimes just observing, watching, and they are just so much fun to see, and they can process so much bacteria.
Hey, so Rich asks, is the presence of rotifers accompanied by a bad smelling substance? I'm going to say often no. Sometimes they might be surviving that nasty smell. I mean, if they can survive in sludge, like waste sludge, I'm guessing they could definitely be present in a really stinky environment, but they don't signify the bad smell. So, I find rotifers all the time in vermicompost that smells beautiful,
Right?
and looks aerobic in every way, not sopping wet. It's just often in vermicast, the worm cast itself will become anaerobic on the inside, and these critters will live there. But yeah, no, it's not necessarily that it's going to stink and there will be rotifers present.
Yeah, and I've seen rotifers in very healthy aerobic soils as well. Usually not in the quantity. Like here, we just saw an active rotifer, and this is a dormant one. So right here, not dormant, but it's resting. It's not feeding at the moment. And I saw another one. So within a very small area in this particular sample, three rotifers, and I saw a lot of other rotifers before this as well. So they obviously populated well in this kind of facultative, going anaerobic, very anaerobic kind of, in the kind of degrees. So they can exist in a lot of different soil profiles. So if I see a large amount of rotifers, like a huge amount like here, yeah, it's probably looking like it was anaerobic at some point, or in the facultative state. But like I said, I've seen them in very healthy soils too.
I mean, the truth is too that anaerobic doesn't always smell bad, and that's kind of tricky.
Yeah.
Sometimes you can really smell an anaerobic environment, woof, it's producing gases already. It's obvious, but um, sometimes you can't smell it at all, and it's just a very temporary, natural, healthy thing that happens in a soil ecosystem or a compost or anywhere there's too much water and food at one time. The bacteria are going to bloom, it's going to go anaerobic, and then the situation should right itself. I don't see a lot of bacillus. Are you seeing it on your side, Brian?
Not there's some, but not a lot. There was more earlier. Like I said, it's about
Interesting.
an hour now since I added oxygen. The biggest thing was the spirilla. Oh my gosh, they were just everywhere. And again, here's another rotifer. I mean,
In an
that's four
hour, the
rotifers.
entire bacterial ecosystem has changed.
Yeah,
that's
Yeah.
wild.
yeah,
Yeah. So sometimes we
yeah.
have people that'll be panicking about their compost. They're like, "Oh my, I found something in my compost that's scary and crazy. And we're like, okay, like give it some compost extract and wait an hour, check it again.
Yeah,
And really,
exactly.
these ecosystems can move quick.
It's almost like it's
Oh, here
after
we
this
go.
giant music
Here we go.
festival and they're
There
cleaning
they
up.
are.
There they
Ooh,
are.
wow.
There they are in great number.
Yep. They're
So
Mm.
hunting in
the
packs today.
so these are spirochetes?
Spirilla?
Spirilla.
Rilla, okay.
Yeah.
Spirochetes are more like they can move all over the place.
Mhm.
Spirilla are like a corkscrew through space. They move
Oh yeah.
Going
and
crazy.
they move at angles.
Is that the one that yeah, it's spiral is that the one that Elaine was like, ah, when you see it, run away?
So yeah, I mean, spirochete and spirilla are both can be scary. The truth about it is that there's not a lot of research like let's figure out exactly all these different species of spirilla and spirochetes and which ones are dangerous and which ones aren't. What we know for a fact is like there are species in both of those categories that can hurt or kill people or animals. And so since we can't tell who this is exactly, yeah, we need to wash our hands, and very
Right.
especially because of the amount. Like look at how many
Oof. Wow, yeah.
spirilla there are in one view. Wow.
Yeah.
Wow.
Holy cow.
It's a lot.
But these
And
can be
you know,
these can be misconceived as good guys, because they're like, oh, I saw this good buzzing and zooming. And it's like, oh
right.
no, you saw a reason to wash your hands. Like
Yeah. And the crazy thing about this is from the same vermi. You haven't seen my vermicompost sample yet, but this is coming from the same vermicompost. And when I look at it, when I pull it from the worm bin, I don't see any. I mean, really, I hadn't seen any spirilla at all. But laying which means it's there. It's just dormant. But when it went anaerobic, it was the right conditions for them to just completely thrive and reproduce. And so it is a good example that even though you can go through the whole process of making really good aerobic material, the anaerobes are going to be there. They're just going to be in a dormancy state. And then they're waiting for their time and say, oh, it's now my time. And then they'll go ahead and replicate and populate. And it's interesting. You can see through looking at the slide, there's areas that had more aerobic things starting to occur. The spirilla are gone. Um, and so this may be just an area in the slide that still maintaining kind of more that anaerobic kind of condition. And I'm wondering with those rotifers stirring things up. And granted, it's under a cover slip, but maybe it's just enough by the side where it's able to pull enough oxygen in. I'm making some hypothesis here, but that's what I'm assuming that's happened. Well, I'm glad I was able to show, and thanks, Keisha, for bringing that up, of having me take a look at this sample because that does provide you some differences there. All
Yeah,
Yeah,
right.
and then you can also see that even in that anaerobic environment, the aerobes are persisting. I think a
Mhm.
lot of people get the idea it's like good, bad. Like as soon as you cross the line, you're in scary town. But truth is, this fungal hyphase, I mean, we don't know if it's alive right now or dead but you can still see
Right.
testate amoeba, you can still see cysts, you can see spores. At this point, it hasn't been anaerobic long enough that the bacteria have consumed absolutely all of the organic material and all the life. That's the bad thing that happens in anaerobic soils. It's you have to like bacteria consume organic material. So when you deprive something of oxygen for long enough, all of it disappears. And that could be cool, Brian. Like, why don't you keep this test tube for another few months? And let's
Yeah.
look and see who's left then. Because I've done this test before, and what happens is like there are some gnarly aerobes. Like some nematodes can last forever. And they just
Right.
start eating everybody, like spearing everybody. Like they start acting different than they normally would. But eventually it's an empty room and there's just a few ciliates maybe ticking around or just some bacteria. There's nothing left. Like it all gets consumed. So it's really fun to watch this stuff over time. I think that's a cool, very adventurous project.
I'll keep it for our microscopy class because I think when we get to lesson 5, we're getting into the other stuff. This is the kind of more adventures type of thing. So that will be in end of January, I think, when we start looking at that. So I'll keep it for another month and a half, I think.
Cool. Yeah, Stacy asked
All
some
right.
weren't moving. She mentioned those ones that weren't moving, and she's wondering if the oxygen's starting to get them.
I don't know. Be honest, I'm not really sure. I think they're attaching to the top of the bottom of the cover slip because that's where my focal plane is at right now.
Mm.
Um, So why they're not moving, I'm not really sure, to be honest with you.
It'd be
What
fun
would
if the
cause them to
rota
stick to the surface?
It'd be fun if the rotifers start colliding with the spirochete area. They're just gonna go to town there.
Yeah,
This is
I
where the
was
adventure
just
gets fun. I mean,
Yeah,
Marie that's what she wanted to share. She's got some
yeah.
video. She's got her awesome samples. She's also got a video of fungi eating a nematode.
Oh, that'll be cool.
It's pretty wild.
Cool. Well, Brian, do you have another little sample you want to share, or should
No,
we
no,
go to
no. I'll
Dan
stop
now?
right now. Let's
Okay,
take a look at samples from Marine from Dan, and then we can come back and look at my vermicompost at
awesome.
some
Let's
point
do
because there's
it.
some good life in there. Let me go
Great.
and stop sharing here real quick.
Okay. Yeah, Dan, how about let's hop over to your sample and we could talk about how you made this, how you made your compost, and what you've been seeing under the microscope.
Yeah,
All right,
great.
awesome.
Hopefully you can see that okay.
Yeah, it's so smooth.
Sample's been on there for a while, so I'm starting to see some movement. But
Nice.
the liquid coming through. This is a 1 to 10 dilution.
Oh, real
So
quick, Dan, any chance we get
yeah.
this full screen?
Let's see.
I know
This
if you
is
do the
on.
window zoom in or like that little
Oh yeah.
magnification, yeah,
Yeah.
150.
Yeah. Okay.
Okay, sweet. That looks good.
Okay. So yeah, it's a 1 to 10 dilution. This is compost that I made in the spring into summer, where it's been sitting kind of combined in a mother pile right now. And so yeah, I just pulled this in or took a sample yesterday. It's under about just under a foot of snow. So it's been thawed out a little bit, but yeah, this is the sample here. Just trying to think where this is 10x, so I could go in unless you see something we want to.
Well,
There
there's
was
lots of
a,
fungi. I'm
yeah,
pretty impressed with is that
Yeah,
fungal hyphae that we're seeing span the entire screen right there?
yeah, yeah.
Let's
Good
look at that.
job, Dan.
Go in there
yeah, good job.
We'd love to see. Oh, there we go. You're at 400x total.
now. Yeah, yeah,
Beautiful.
And then you've got that fungal spore right there is really beautiful as well.
Yeah.
Wow.
yeah,
Are you using you're using an LW microscope?
yep,
Yeah, yeah, this
You
looks
on the
yeah.
great.
It's
I4?
Revelation 3.
Nice.
Revelation
Version
3.
three. Nice.
What camera are you using? Minivid?
It's a yeah, mini vid. Yep.
I mean,
All right, I'm gonna use, I
nice.
have that microscope too. I'm bringing that to the next session. This looks nice.
That's
Yeah,
right. You
this
got
is
the
great.
mini camera as well, right,
I
Keisha?
do, yeah, I just don't ever
Yeah.
use it.
Yeah.
And that's coming across nice.
I love
Uh,
it. Yeah, this is
Mm,
great. Super clear.
Yeah, so many spores. Spores, spores, spores,
yeah.
spores, spores.
Was trying to find some of there was quite a few testate amoeba,
again,
Arcellus,
how
and
old was this compost, Dan? I'm sorry if I missed it.
it's from the spring, summer. It's kind of all been combined into one mother pile, but
Oh, that's right. You said that.
within
Yeah.
this last year. So
So it seems like it's almost a 6 month plus old pile.
yeah,
That's
I'll
great.
zoom back out a little here. See if others.
But yeah, see some amoeba.
Yeah.
Yeah.
See.
Oh, there's a guy. Oh, nice.
We could test any right there.
Amoeba friend.
Wait, you think that's a spore right there? Would it like right next to the testate amoeba?
I think so. It looks kind of
Right?
like it. Yeah.
It's all folded. But I was gonna say, Ellen asked, are the spores the round reddish things, or dark or reddish brown, round? But spores can come in all shapes and sizes and colors, and often they're round, but they can sometimes even look like that little folded guy next to the testate amoeba.
So Rich asked about naked amoeba as well, how you spot naked amoeba. A lot of times
Yeah.
you got to kind of train your eye. You're looking for kind of like a blob moving. I did see naked amoeba in my vermicompost sample earlier. So if I get a chance, if I find one before the end of the webinar, I'll show it to you guys. They're fun to watch though. They are just super cool as they just ooze through the soil.
Really good work, Dan. There's so much fungi here. There's so many different sizes and shapes and colors of testate amoeba.
Yeah. Mm-hmm. 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 Mm-hmm.
Tons of fungal spores, good aggregation.
Yeah, this is great quality compost.
And you
So
started
this is where we
sowing.
would go through and assess, right? We like, okay, we look at this compost and go, yeah, this would be great inoculum, making
Mhm.
compost extracts or compost teas or use it in your planter beds. You would expect if you're using this on a real poor soil to use as a tool to start to rehabilitate, to inoculate that soil with a microbe. So fantastic, Dan.
It's interesting that the hyphae is showing up. It looks like a fiber optic cable or something.
Yeah.
It does, doesn't it? Yeah.
I think it is. I'm certain that's fungi,
I,
though the tip is so like.
Yeah.
so broken off
Somebody asked, "What's the
straight. Yeah.
zoomers?" Can you pull it
Yeah.
into? Oh, there's your zoomers.
Yeah.
Yeah. I think it's just some kind of, modal bacteria.
That's what I was thinking. And
They're pretty tiny.
yeah, I've asked
Yeah.
Keisha a couple times, I think, about those because I see them a lot. With they're
Yeah.
not like the bumbly flagellate type style, but they zoom around and but
Greta's asking, "Is this vermicompost? This is your thermophilic compost, right, Dan?
yeah, right. Thermophilic
This is a
compost.
compost Dan made in the spring. Oh, you
Yep.
just said it. I was focused on
Yeah.
the
No,
Yeah.
yeah. In the spring, this spring.
Yeah, and someone else asked, like, what happens during the winter? Like what's going on with the microbes when there's snow on the ground? Like, do they just disappear? Like, and I think you can see here that some of Dan's bacteria are quite active, although they've been warmed up for a little bit inside.
Mhm.
Um, Fungi is still growing in the winter. There's still microbes happening. When you get under the ground, it's not as chilly as it is up in the air. So there is still some work happening with these guys when it's cold. What is it? That's cool. It's like a
It looks
testing
like a
maybe that
test
got,
amoeba shell
Yeah.
that's kind of broken a little bit.
yeah,
Yeah.
Yeah.
interesting.
Spore, fungal spore there. I always
Well,
wonder.
I'm impressed, Dan. It's looking good.
Good,
Yeah,
good.
that's great.
And you've been using the compost and compost extract. I'm sure you've been making extract with this compost.
Yeah, yeah, we've made a lot of extract. That was one big thing coming out of the Compost Academy as I kind of accelerated into my extracts and we've been able to apply them. We saw a lot of
Good.
great results this summer in our garden beds and
Awesome.
garden boxes and other growing
Have
areas.
you been
So it's
looking at your
been
garden soils to kind of track those over time, just kind of see how they look?
um I have like we
Yeah.
applied the extract several times in the summer and I kind of monitored it and the soils looked great. They looked really good after applying the extracts. So
That's so cool.
Yeah,
yeah, I don't have a
fantastic.
sample of that now, but yeah, it's been
Sure, sure.
yeah.
Stacy, oh, there's a few questions that came through. Okay, first, Rich asked, how much life is smaller than what we're seeing at this magnification? And like, I think a whole lot, Rich. I know
Sure.
like there's archaea are smaller, some bacteria you can't see,
Versus
viruses are like, yeah. This is we're
super
just like
prevalent.
we're like basically just scraping the surface here. And
Yeah.
and you can go in, we can go in further. I know Matt Power students are often in like they are loving the is it the oil immersion or the non-oil
The,
immersion?
probably the dry 60x or 100x
Yeah, where
objectives.
it's
Yeah.
really going further into magnification. But for me, I like this. I like looking at like 100 times what the eyes can see, 400 at most, just because the clarity is still
Mm.
so beautiful. You need to get into like really expensive microscopes and processes if you want to see the teeniest of the lives. And then Stacy was asking about that broken amoeba. Would you count it in an assessment? I mean, we're talking soil
Mm.
food web school here again, Stacy. So
Yeah. Yeah.
I'll tutor you on that. I wouldn't count it if the shell was broken. But
Yeah.
if you were to count it, it wouldn't be the most horrible thing. Even when we're looking at SMAP, it's a guess. You're really trying to identify how many amoeba am I seeing, and that one was pretty big. I mean, you could take the way of saying, "Oh, that was a huge amoeba. I probably broke it with my cover slip, and then so it was probably alive, so I'll count it."
Right.
Or you could say, "Oh, it looks damaged. It could be dead. I'm not going to count it." Um, that's the reality of a microscope assessment that has to do with human eyes is you just have to make the decision and trust yourself and be okay with it. Ooh,
Oh, there we go. There's an
Nematodes, huh?
nematode.
Yeah, it's moving pretty quick. Hasn't slowed down much, but there's been a few in
Were you able to
here.
identify it earlier, Dan? Do you know? Or
No,
it's
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 never, yeah, it's never it got to where I could identify. I've seen mostly bacterial feeders and a
Your
lot of
compost.
this, yeah, like, but trying to see if it'll slow.
Sometimes you can get just enough to be able to see like the cuticular valve will pop pretty
Yeah,
easy. And so that's
yeah.
usually a good sign. I'm not seeing it. I haven't seen it really yet here. Um, and then, a lot of times when we look at like nematodes where we're trying to assess what they are, kind of use the morphology to figure it out, you might have to heat stun. And when you heat
Hmm.
stun, you have to heat up the underside of the microscope enough to be able to stun it without actually damaging or killing the nematodes. Um, I use something like this, a torch, to be able to do it. And it takes some technique though. You have to practice it, and you just
Yeah.
heat up underneath the slide, and it will stun them into silence, really, or no motion.
What a great sample. Thanks, Dan. That was
Great.
awesome.
Yeah.
Yeah.
Well,
Beautiful.
Should we take a look at Marie's?
Yeah, let's check out Marie's and then we can hop on to some other things.
Okay. I was waiting for a long time.
Oh, did it
So
dry out? Oh,
yeah,
bummer.
but let's see. I have one right there. I am in 10x. Let me, can you
Hey,
see
Marie,
there?
while you're doing that, I'll give everybody kind of a little bit of a tip. If you ever want to keep your slides, especially if you live in a dry environment or use air conditioning, I just have a glass lock container with a wet paper towel in it, and then I put my slides in the container. And when I'm not using them, I put them back into this chamber, and it just keeps a nice amount of humidity. You can get the slides to
Uh,
last for a couple of hours in that way.
Wow, great tip.
Yep,
Yeah.
no problem. All right, let's take a look here. What do we got?
Okay, so what we have there is an archilla,
Yeah, look at that.
and
Oh, oh,
I will,
first of all, Marie, what magnification are you at?
yeah, I was on the 10, but now I am, can you see there? No.
It's a
It's
little dark. You
Wait,
might have to
dark.
increase your
Yeah.
light source a little bit.
yes, there,
Yeah, there we
There
go.
we go.
Much
That's way
better.
better. Yeah.
there. So I have tested some EVAs here, but this one is an archilla, and I can see this one because of we can see the mouth, which is right there,
Hmm.
and it's a beautiful one. Here we are in 400x, and I have seen others in central Chile, which are full of crystals, and they look like princes, full of jewelry.
Right.
But
Yeah,
here they don't have so much because this is a compost, so no much minerals to decor themselves.
sure.
And
Um,
yes, sir.
so there was a question just popped up from Levin about he missed the explanation about how to keep a slide. So I don't mean to interrupt you real quick. I use a container. This is a glass container, and I have a wet paper towel in here. And then I have my slide sitting in the very bottom in here. It just creates, it's a humidity chamber, really what it is. So your slides will dry up much, much less frequently. All right, Marie, sorry to interrupt there.
Yeah, no, it's okay. I needed the explanation as well. Here I have a nematode.
Oh, look at that.
Wait a little bit to make it. Okay, so this one is not dead,
Just sleeping.
but static.
Ah, there we go.
So,
Let's take a look at its mouth and see what we can see.
yeah, so let's see the mouse. I think it is an omnivore. Omnivore.
Oh yeah, right there. So you can see that kind of wide, shallow mouth.
Yeah.
That's kind of what I'm seeing. Do you have a cursor you can kind of put across that to kind of show everybody what we're talking
Okay.
about?
So
Right there,
this is
yeah.
the mouth, and
Yeah.
it is wide. And sometimes I have the feeling that it could be a carnivore because it has something here. And this is wide and straight. So sometimes I have my questions here if it is an omnivore or a predator.
Predator.
Predator, yeah, because of this little
Yeah.
thing. But I don't see the tooth, but I see this little thing.
Tooth.
But
And
then
usually the tooth is pretty prominent.
Yeah,
yeah.
I mean that's the truth with nematodes is the deeper you go down that rabbit hole, there is no like very strict defining characteristic of who eats who. Like predatory nematodes don't necessarily have a tooth. Sometimes they have a spear, and so it's interesting when when you actually see them eating each other, you can see, different mouth parts eat different things. Some fungal feeders actually eat nematodes. Some omnivores might be able to eat higher level organisms that you wouldn't normally
Oh,
think. And you can especially see it when you look in water systems. A lot of the times you can see what's inside the nematode, so you see what it's eating.
hmm, wow.
Hey,
Yeah.
Keisha, there was a
Well,
question that
there.
came in from Seth. It was, in my limited experience, one big challenge has been finding photos, examples of all the different microorganisms online. I seem to only find the same few examples. Is there a place or way to find some sort of key or encyclopedia of microfauna with photos and functions? This is a good time to plug the microbe library, right?
Yeah, sure, Seth. I mean, I plug it and apologize at the same time because I use social media just because it's easy. Um, we are creating our own, we're creating this resource for sure. I've been working on it for a long time. I have thousands and thousands and thousands and thousands of photos. Um, but if you go to Facebook, I have a group. You have to search it out and find it. It's called Soil Microbe Library. And there's actually two. There's a group and then there's a page. So you could follow both. But on Facebook, what it is there's probably 5 or 6 thousand, maybe there was like 5,000 people a few years ago. And a lot of them are Soil Food Web students or they work with KNF, they work with microscopes, right? Everybody there's a microscope nerd. And they share photos, they share images, and we all commune and talk about what we're seeing. And then on Instagram, there's a page as well called Soil Microbe Library. And it's a little bit different in the way that people send me images that they don't know what they are, and then I share them and talk about them. But there's a huge history on both of those. You can just cruise through and look. But there's a lot of other really nice pages as well. If you want to look for protozoa, my favorite is Arcella. I'll actually type it in here. Um,
.nl, right?
Arcella.nl. Oh, no. I put in Marcella.
Wow, look at this. Look at this right in the middle of the frame. That looks like a wild branched fungi. That's really
Fungal
cool.
weft. Look at that, Marie.
Yeah.
Beautiful.
So let me go to the 400.
So Marie, is this a compost you made in your backyard? Is it a vermicompost?
Yes, this is
Okay.
a compost. There are two kind of testes amoeba there. It is a compost that I made in May, which is our autumn.
Hmm.
It is a thermophilic compost, and it had lenga bark, which is a native tree. It is a kind of notophagus. Orchard, mainly brassicas, because that's what it grows outside here. A horse manure with some shredded wood that I took from the rodeo place very close to my house. Some hen manure to make sure that the temperature will go up because we were starting the winter. This was in May. And then it has been sitting there after the thermophilic uh, doing it, right? It was sitting there like 6 months, and it is in a bin, which is a wood bin, open. Here, the wind is very big, so it's kind of dry on the top because of the wind. Sometimes we have up to 140 kilometers per hour. That's many miles. I don't know many. I don't know how many. And it's very strong wind. And also it has been sitting outside under the rain. So the under is more wet and the upper part is dry because of the wind. So this is what we have in front. And it came quite well.
Looks beautiful.
So what we are looking there is two testate amoeba, and here, which are these long ones, and here I can see another one that is reproducing, probably a different kind, probably. Like, I will
Can you
try
focus
to,
on the fungal hyphae a
yeah,
little bit?
yeah. Okay, so now we go to the hyphae.
They're on a totally different level.
Yeah.
Wow.
Wow.
And it has, yeah, we can say it's a beneficial fungi because of the color and the straight walls and the septas. And yeah, it's really beautiful.
Yeah, I haven't seen
It's
that
super
very often.
beautiful.
It's, you know,
Yeah,
like a fungi tree branch in a sample.
yes,
That's really
yes.
beautiful and rare to see.
It has been sitting there for a long time, nobody touching it. I mean, 6 months not touching. It has some clamps there. No, it's no clamps. Yeah.
Marie, when you see
So
that under the naked eye, when you look at it, do you see a lot of the mycorrhizal like fungal strands like with your
no,
eyes?
not
No.
many. No, actually not.
Yeah, it's actually crazy, Raleigh. The guys at the lot used to try to get me like the most fungal samples, and they'd pack them all into all of the fungal hyphae into a bag. And those don't get the best. Often it's like what you can't see that
Mm.
it's crazy. I will warn Marie, we can't say for certain that it's beneficial fungi just because of the color and the diameter and the septa, just because there are certain species that are parthenogenic that do have septa and do have color, and there's not very many, but we got to be
Mhm,
careful with our words and say it's
mhm.
most likely beneficial fungi
Yeah, that's true.
because it could
That's
always be
yeah.
not so
I
beneficial.
am always I my head is full of doubts.
Yeah, good.
Yeah, actually, I have more questions than answers.
Isn't that
But
this
yeah,
work?
I assume it's a nice one. I like to think it is a good one.
Yeah, yeah, it looks beautiful. I would consider it a nice one, but when we say we know it's beneficial, that's where we get into trouble because we really don't
Yes,
know.
right.
We just have
It's
to be
true.
careful with the language.
Yeah, yeah, it's true.
Look at your test
And
but that one was so it looked like ornamented almost. Like a
yeah,
glass. Yeah.
well,
Oh, that one's pretty too.
yeah. I don't know if maybe I should show the videos that I have of the nematode. I mean the fungi eating nematodes.
Oh, that'd be
Please.
great.
Yeah.
But I think real quick, Murray, before you do that, this is a great opportunity. We're at about an hour mark. I could tell people about the microscopy program, and then we can go back and share some of those videos. So let's
Perfect.
Okay,
hop on
whenever you.
there, and
Yeah.
then I'll share everybody. Okay. All right, awesome. So for folks not acquainted, we have this really wonderful program coming up called the Microscopy course through Compost Academy. So this is we meant to create this to make it be a really practical training that's really effective to show you how to use microscopy effectively to identify organisms, to see what's happening in your soil, to ensure you're on the right track, and to do this with a group to get training professionals. So it almost feels like an apprenticeship in a lab. So you were going to be going working with each other week by week for about 6 weeks through a 6 weeks guided microscopy training with Keisha and Brian. We're going to have different lessons each week, along with self-guided video lessons about how to use microscope, how to use samples, how to do qualitative analysis, which is very important. And we're going to have an online community with challenges and feeds and things to help us out along the way. I think one important distinction about this, a lot of times we get bogged down in things like SMAP with quantitative analysis, and this program is really showing you how to do more fast and effective kind of in-field qualitative analysis. So that when you're looking at samples, you can tell if you're on the right track and not get really bogged down in analysis that takes hours and hours and hours to do per sample. And so this program, it starts December 11th. There's 6 weeks of training, self-guided video lessons that are week by week, and online community and challenges. And all this together, it is $725. And if you're a student of Compost Academy, you get $125 off, which would bring it down to 600 if you're a Compost Academy student. And a little bit about the format, yeah, live group coaching sessions, which you'll be a part of, week by week training. We're all hopping in this in together, kind of like this format, but each week it's a different topic that'll really get you in-depth training. You'll get those self-paced lessons that'll guide you, help fill in your gaps of knowledge. You'll get challenges to help find samples, find different organisms, and really make sure that you're on the right track. And of course, we're all doing this together in a community where we're going to share feedback, get questions answered. So, and most importantly, you're not doing this alone. It's like imagine if you're spending hundreds of hours alone in a microscope. It's boring. It's kind of lonely. But doing this together, you're part of a group of very experienced microscope fanatics and learn together with expert level training. Oh, sorry. Open up that line. And the schedule is we start December 11th, the foundations. We go week 2 is December 18th. We really dive into the techniques. Week 3, January 8th, we started really diving into the organisms. Week 4, we kind of wind up more with bacteria and protozoa. Week 5, we go higher on the soil food web chain and dive deeper into nematodes, arthropods, and more. And then week 6, we really dive into the qualitative analysis, doing soil comparisons and student practice, and learn how to effectively use that when looking at samples. And also want to give you guys this, because you're joining this webinar, we have a special deal that's only for people here. If you signed up with this link, you're going to get $100 off. And I'm going to post this right now again in the chat. This is only for folks who attend this webinar. And this is, you'll have this price available till the 11th. But we really encourage you to hop in. There's no great opportunity to really, if you've been kind of wanting to dive into microscopy, there's no better time than this. Like I really wish I would have had this when I was learning microscopy because I had to go all the way to a lab and pay like, thousands of dollars of my own money to do this lab work. And yet I would have but having this training with someone like Keisha and Brian is awesome. Like there's nothing, no comparison to it. Like just the amount of knowledge and shortcuts you get by learning one-on-one with them and with a group, it's priceless. So I'm going to post that link and we can get back to more samples. But we hope you guys join in. This is going to be really fun and it's going to like really make you fall in love with microscopy. And if you're already a microscopy expert, you're gonna level up your training to a whole nother level. So this is going to be a really great 6 weeks. All right. Well, I'm going to post that link
So
and we can share some videos.
Hey,
I
Raleigh.
want to share.
Some
Yes.
folks are asking if you know what time that we're going to be doing it yet.
Yes, we are scheduled for 1:30 Pacific Standard Time. That would be 2, 3. So that would be 1:30 Pacific time, 4:30 Eastern is the time that it happens weekly. And there, of course, there's that gap in the holidays, so you don't have to worry about like we know everybody's going to be busy on a holiday break, so we don't have to worry about like, oh, I don't want to attend on Christmas or New Year's. Like we've
We
planning
take
around
two
that.
weeks off, yeah,
Yeah,
through the holiday time frame.
Yeah.
great.
Stacey
So,
also asked, is there going to be time to help understand how to set tube view settings? And I'm guessing all those technical things. And yes, we thought about that already. We have a bunch pre-recorded for you, like the basic information, so you can always go back and watch that over and over again. And then we also have time scheduled for one-on-ones or five-on-ones or whatever. We're assuming that a lot of the students are going to have issues setting up, and so just like we do with our normal Compost Academy, we're going to have the ability to, go to the side, take a little bit of extra time that's not necessarily our weekly, just to make sure everybody is got their microscope working. So we'll do what it takes to help you out.
Yeah, we have already planned for that first week. We'll probably do two meetups, which are going to be not part of the live coaching, but it's a scheduled time frame. We'll do some different times, zoned kind of scheduling, so people can just come in. It's like office hours, like if you're familiar with college, come in and you've got issues with your microscope, set it up, that type of thing will help you kind of through that process. So
Mhm.
great. Um,
Do we want to look at that
I
video
think
of
let's
the
do it. Let's check it out, the nematode
yeah.
trapping fungi.
And then when
great
Marie
video?
gets that, I got some great nematode stuff to show you guys after that
Whoa,
too.
Ooh,
Can
cool.
you see that?
that's
Yeah,
a
we can
good
see it.
one.
That
That's a
Okay.
really good one. Wow.
that's a great one.
Jealous of your
Okay.
compost, Marie. You've got the best.
Yeah.
Yeah. But see, it's the same compost I showed it, but 1 month ago.
Yeah.
So I found this. It has some rings, and this nematode it's dead and it is trapped. So I'll play it now. So I am looking at how it grows, and it grows quite like an Oomycetes fungi
Hmm.
with bubbles, and it
Mhm.
makes some rings. And that's where the nematode, and it is a very big nematode, was trapped and died.
Isn't that interesting? So this is I mean, I would consider this a beneficial organism, right?
Yes,
But
yes,
it's
Yeah.
clear and colorless and it's got bubbles and is it fungi
yes,
or is it omycete? And omycetes aren't fungi, but there
no.
are fungi that are clear and colorless and kind of messy looking.
Yeah.
This is where the soil food web world gets a little bit complicated, and this is why exploration is so important. Just being able to like, yeah, Megan, this isn't an omycete. I don't think this is a fungi. So that's the point. Is like omycetes are clear, they're colorless, they're messy, they're not necessarily skinny. Sometimes they're huge, like
I
very
have seen very
thick.
big ones.
And
Yeah,
some fungi
Yeah.
is clear and colorless and messy.
yeah,
It's
yeah,
there is not a good and bad. We've got to get away
yeah,
from that. There's no good
yeah,
guy, bad guy.
yeah.
There's diversity, and that's good. You know, seeing all of like the fact that we looked through her compost and saw so many different shapes of fungi, so many different shapes of testate amoeba, lots of nematodes, and just flagellates zooming around. That's what we can trust and say is good. Like, wow, this is a great ecosystem. And this is a lot the way that we're trying to run this class is to be not so specific about who is this, are you good or bad, but more, this is a walk through nature, and we're trying to say, oh, look, it's a bird. Look, it's a worm. We're trying to become a little bit more basic and just familiarize ourselves with all these organisms, who they are, where the gray areas are, so that we can have a more robust idea when we do try to sample, when we do try to like, run an S map or run something numbers. So yeah. And then also omycetes could do that, right? There are probably omycetes that kill. I don't know. I've never heard of them, but there could be omycetes that are
I don't
absolutely
know.
predatory on insects. That's something to look into. I think I'll put that down as a note, actually.
I'm trying to, can, I'm not sharing that, right?
Mm-mm.
Oh, not yet. Yeah,
No.
if
No, not yet. Right.
Marie, to do that, you do share,
Yeah,
advanced,
yeah,
and then you would select the video file.
right.
Yeah, it's
Yeah. Okay,
popping up
this
right
is,
now.
now it's there? Yes.
Yep, yeah, we can see it. So what is this we're
It
seeing?
is the same, it's not the same one, it's another one, but it's the same kind.
Okay.
Okay, there. See the rings, how it is formed, how they form? Yeah, I didn't know if it was an Oomycetes or a fungi. I have no idea, but I was lucky. I didn't encounter them when I was doing this map, so I didn't have to decide. But I was
Yeah.
wondering,
And
yeah.
it's interesting too. There's some, so we use different species of fungi that we get from labs. So Bavaria bassiana is one, and it's confirmed what it is. But if you look at its hyphal structure underneath a microscope, it looks very much like omycetes, very, very much. And
Mm,
the reality is like I don't. There's a lot of issue with us feeling like we know so much. It's so
yes.
important to keep an idea of curiosity and open mind. Because even with animals, right? Like, how often do you see an animal? You're like, well, that looks like a deer, but hey, that's not related to a deer at all. It's a different animal that looks like a deer, but it's not. So we need to not be so certain that we know and just stay open because this is super cool. Those really do. Like, have you seen the electron microscope image of the nematode trapped in the ring, Marie?
Mm,
You should I'll
no.
try to you should search it out online. There's this famous and Elaine used to show it all the time. That looks like a live image of
Right.
just the ring, you know?
That does,
Like a
isn't
snare set or something. It looks like a
it?
like a
Right.
trap.
Yeah, that's cool.
Yeah, I have another one. Let me find it.
And if you have that water bear sample, the tardigrade,
Yes,
that'd be really
yes,
fun too.
yeah, okay.
Oh yeah, I didn't upload anything really, sorry, I
That's
forgot.
all good.
We have
Sorry.
a question here. What is omiscetes? What is an omicete? It's so that the people they used to be classified as like a pathogenic fungi or a type of fungi. But now I believe omiscetes are characterized more like protists, if I'm correct. And some of them
Mm.
are pathogenic to plants, and some of them are not. They could be beneficial in soil. They could be, inert, but they can show you that there could possibly be pathogen in the soil. So when we're looking at the morphology of the organisms we see, omycetes would typically look kind of messy, not so structured. And in a way that, this looks like that, messy, bubbly. You'll often see their spores right on the hyphae.
Can you see that?
Yeah, see, it's got a real omycete look. Really, really.
Yeah, it
Yeah,
It
does.
makes me want to
yeah.
deep dive into it and see like how many omycetes are actually that are known
Yeah.
trapping nematodes.
Marie, what were you looking at this sample? Was this a compost sample or was this a soil?
Same compost sample, but just 1 month ago.
Ah, wow.
A
So
great
different.
sample. It's so fun.
Yeah.
Yeah.
Yeah,
It's
a lot of
very
diversity.
interesting.
It has right there, it has a nematode. I don't know
Yeah, you can see it. It's eating it.
if you
Right.
can see the nematode, but it's very messy, full of rings, and this nematode is huge. I don't know why I cannot make it bigger. Can you see it?
Yeah, I see it. It
Hmm.
Yeah, we can
looks
see
coming
it.
in fine for me. Yeah.
Yeah,
See
yeah, I
Yeah.
it
see
for
it.
full screen? Yeah. So that's where a huge nematode is in there.
That's crazy.
Ye-
It's almost like a lace of traps.
Yes,
Like good luck getting
yes.
through that.
And that's the
Yeah.
tail.
Yes.
Danger zone for nematodes.
Yeah.
Right.
Nematode
It is a
minefield.
nematode.
Avoid.
Yeah.
And Stacy, yeah, Stacy asked about Tube View. Absolutely, we can help you with Tube View settings on there. I know, like I've been experimenting with it, and there's fun tricks you can basically use to hack Tube View. I know when I record videos, you don't actually use Tube View, you just record your screen, and you get a
Right.
way faster frame rate. So we could show you things like that, especially if you want to record microscope reviews. We'll show I can show you how to do that. It's really fun too. It's like to get good video of a microscope of what you're seeing under the microscope and share it around. It's a really good resource. Especially it's like I imagine if you're working with clients, they'd probably want to see that too. Like really good sample footage.
Yeah. And now the last one is the tardigrade footage that I have,
Oh,
but my
excellent.
computer is a little bit slow. So
Oh, no worries. Well, maybe while we're waiting, Keish, you had that really good story that you actually went down to Apricot Lane. It's Apricot Lane Farms that where they
Oh
filmed the
yeah.
biggest little farm,
Yeah, I got to
which in
go.
the film is like, oh my gosh, it's so epic. And yet you looked under a sample and you're like, it's got room for improvement.
It does have room for improvement. What I'm excited about their wear tested amoeba, I was hoping to
Oh,
put it up today, and because I didn't get to do a representative sample of the bag, I just grabbed some. Um, but I think the truth is, sample could have been in the bag for like a year from
mm.
what he
Yeah.
said. I don't think that this sample was like, or maybe the compost itself was a year old. Oh, look at that.
Hey, there we go. There's our friend.
yeah.
Hey,
So
buddy.
this is the mouth, right?
Hmm.
Right in here, the mouth. This is maybe the pump to suck. And these are the little how you say, how you say in English? Nails.
Pause. Oh
Pause.
yeah.
A clause, yeah.
Yeah. Yeah, see the little ones there? Three in each. This one is not moving, but we can see it, the whole thing.
I'm just taking a nap.
Raleigh, can you
Yeah.
add the dates of the microscopes?
Yes, for
Yeah.
sure. And yeah, Franz is asking about he's New Zealand time. So let me see, 1:30
Okay,
p.m. to New Zealand time. I'll do that conversion for you.
that's all what I have. Thank you very
Very
much.
Thank
good.
you, Marie. That was
All
You've been
awesome.
right.
exploring. Thank you for
Yeah,
sharing with us. That's so cool.
yeah.
It's a really good shot of the tardigrade as well. You can't always get the claws and you can't
Right.
always see all the parts because they flip around. Sometimes there's upside down and all you see is their
Yeah,
claws. That's really
yeah.
nice. Thank you.
Thank you.
Yeah, so friends, if you're joining us from New Zealand, it's actually great news for you. 1:30 Pacific time is 10:30 a.m. New Zealand time. So if you're coming in from yeah, if you're in New Zealand, seems like it's a really decent time frame that you should be able to make those classes. I know for folks who want to join from Europe, it's a little bit late, especially in the Netherlands, but that's why, we do a lot of self-paced lessons. You could always hop in on the and see the replays, and you can do one-on-one consulting with Keisha and Brian, where they could do like one-on-one microscopy training and kind of fill in the gaps, where you can hop in on a one-on-one call with them. So I'll put that in there.
Hey, Brian, there's a question.
Yeah.
Um, and since you've got a nematode right there, should there be a good free living nematode population?
Yeah, interesting. Free living, I guess, is maybe a somewhat of a subjective term. I mean, we want to see a lot of diversity of nematodes in healthy soils. And in fact, I have a really healthy soil where I have an Asian pear that's doing fantastic, and I see predator nematodes. I've seen bacterial and fungal feeding, but then I have root feeding nematodes in there as well. Does that mean that the soil is danger zone and really problematic? No. I mean, having a good population, a good diversity is quite all right. I have a client of mine though that has a bunch of diseased olive trees. And when we look at the nematode population there, it is chock full of root feeding nematodes. And so the diversity index is off. And so for me, diversity is kind of the key thing I'm looking for. Um, and just like when I see compost, if I only see bacterial feeders, that's great. I want to see them in there, but I also want to see fungal feeding nematodes and predator nematodes. And I think my compost is going to be a much better compost if I've got that good diversity that's in there. I don't know if I quite answered that question or not, but
Yeah, I'll
that's
keep
the
going. I mean, I would say like
yeah.
the free living nematodes. I've heard that as a term to describe nematodes that are not endo or ectoparasitic. So
Hmm.
bacterial feeders, fungal feeders, omnivores, predators. I've heard that described in literature as free living, where like the endo and ectoparasitic are like either living inside or outside. But I do, I tend to I like to look at nematode populations as an indicator for healthy soil just because um, I I don't think they tolerate disturbance very well. I don't think they tolerate chemical loads very well. I think when you see a really nice, diverse population of free living nematodes, including potentially parasitic, um, that it can be, that's a really good thing. I'm happy when I see a fungal feeder and a predator and a omnivore and a bacterial feeder all in a soil sample. That's incredible. And I know for our compost, ever since we've been reliably able to find the predatory nematodes. I feel really good about that. Just sending it out the door, knowing that some of our nematodes snack on other beasties, it feels good. I don't know what they
Yeah, no
eat or
doubt.
who they eat, but
Yeah, exactly.
yeah. He also asked,
So
does the diversity change dramatically depending on soil moisture and temperature? And yes.
it does for sure. So the sample we're looking at here is from a vermicompost, and right now I'm zoomed in at 400x total magnification, and I'm thinking that this is actually a predator nematode. Looking at that mouth part, It's hard to see.
Hmm.
You guys see it okay? Is it
Mhm.
coming across clear or not?
Oh, maybe we'll get lucky and it'll eat this nematode right here.
Whoa, two of them.
Yeah.
I have video of a nematode attacking and eating another nematode. Casey and I in real life, you can't hear it on the video, but we were going, is that a predator? Is that a predatory nematode? Oh my gosh, is that a predator? And then it like grabbed the other nematode and we were like, oh, it is a predator.
They're
It
nematode.
definitely is. You couldn't tell by the mouth part though. Its mouth part it looked pretty big, but kind of like this one,
Yeah.
right?
This
You're
mod
like,
part's hard to look at.
uh, it also keeps coming directly towards the camera, so you're looking at its mouth wide open from the opening.
Yeah.
And that's a fungal feeder, yeah? Or no? Oh, maybe that's a
The other one, I think it's an actual omnivore. Look at the mouth. It's a little bit.
okay, I see it now. They're moving so
Oh,
fast.
oh,
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 If they
oh,
start
oh,
eating
oh,
each other, that's going to be
Wow.
wild.
yeah.
I
A little battle of nematodes going on here.
know. That's crazy.
is
It's
this
so
your
wild.
soil or
It's
your
such a
compost, Brian?
This is the vermicompost.
Beautiful.
Yeah.
it's so cool seeing the difference, like even in this class, like the thermophilic compost samples, they seem, like they're quieter. You see the amoeba, you see the fungi. It's kind of like, it's stability. And then, this vermicompost samples, it's still this big, microbial balancing act happening. And yeah, just the life you see in there is so cool. Oh, Margita, raised her hand. Let's unmute. Hi, Marietta. Let's see. Let me know if you can unmute your microphone. I okay. Well, you have the permission to speak. I know your hand was raised, so feel free to unmute your microphone if you want to ask a hand raised question. And same thing for anybody here. If you want to do a live question, just do a little hand raise in the chat below, and then I can unmute you because we'd love to hear your voices. And again, this is so cool that so many folks that joined us in Costa Rica that did the Compost Academy are hopping into the microscopy program.
Mhm.
It's so fun. I know like Dan's there, Domingo's there, um, people like Anna are there, Cheryl is there,
We got Gordon coming this week.
Gordon's coming. Yeah, it's so cool just having this community that like, we met up last November and still we're together like a year plus later. It's so cool. And to see each other's like results in the soil is fantastic. Like that's what this is all about. We want to help you from the second you start to like whatever you want to, your journey, a year, 2 years, 3 years plus. This is so great. All right. Geez, what a wild sample. That's great.
Yeah, I'm really happy with the way my vermicompost is coming out.
Yeah.
It's got a lot of life. And in fact, if you look here, I'm not sure if you can see it well enough in the 100X, but there's a lot of flagellates that are moving around. Tremendous amount. You can see them
Hmm.
all kind of right in through here.
Oh yeah.
This sample is just chock full of flagellates. Right now, I'm at a 100X total magnification, just so you guys know.
Hmm.
That's
Do you
beautiful,
do you
Brian.
do you add anything like crushed eggshells to your vermicompost or any of
Um,
the little minerals?
I don't use crushed eggshells. What I actually use is crushed crab shells.
There we
So
go.
I take crab shells, I put them in the oven, I dry them out, and then I put them in a food processor and blend them up until they're powderized, essentially. And then I add the crab shell mix into my vermicompost bin. I probably put, I don't know, a quarter of a cup every 3 weeks or so into the worm bins.
Very cool.
It looks
You're
very
not the first person I've seen do that, but it's so cool seeing. Well, that's a
Well, I live
great,
on the ocean, and crab is really abundant here, and we eat
yeah.
a lot of crab. So it's a great leftover resource.
No, that's great. No, I know like when I saw somebody doing it the first time, I was like, oh, the secret ingredient to vermicompost.
Oh, this will be a good one. Take a look here, guys,
Okay.
on this.
Ooh.
Well, first of all, can anybody tell me what kind of nematode this is? I don't know if I'm zooming in well enough yet.
Yeah, you got it for a second.
Well, I don't see the nodules or the like a big spear. So I don't think it's a root feeder.
I'll dispel the suspense. It is a bacterial feeding nematode. And if you look about a third of the way down, there's a cuticular valve. It's right in this area right here.
Mm.
And watch, it's pumping away.
Mm.
You can
Mm.
see it moving away, just pumping away as it's consuming and eating bacteria. You
Its
guys
mouth
can
too
see
is the
that?
perfect little V.
It is, yep.
It's like a tiny.
There you go. It's got that skinny, narrow mouth. So yeah, I'm happy too. I mean, a lot of nematodes, a lot of good diversity of nematodes in the vermicompost as well. Oh, look at that right there.
There's a bumbleman.
I
Hello,
think that's
friend.
actually a cilia that's getting moved around because I can see the cilia. You guys can't probably see it from the camera, but that is definitely a cilia.
Gosh, what a good, this is such a good nematode extract sample. The
Yeah.
super
Actually,
nem.
I should, when we do our nematode extract, I probably will make an extract of this, a nematode extract
Oh, that'll be great.
of the vermicompost because there's a lot of good guys in here.
Yeah, and I have to do a trade. I'll have to trade you like a bunch of my mother pile compost for your vermicompost filled with nematodes.
You
Do
got
you
it.
not
In
have
fact,
nematodes,
this is a
Raleigh?
really good thing. Say again, Keisha?
So does Raleigh not have nematodes?
No, I mean like when I did the sample, I only saw like one, but
Mm.
but taking his nematode extract from his vermicompost to put in my thermophilic compost,
Mhm.
that's what I want to do.
Yeah. Do you have worms in
Yeah.
your compost?
I do. Yeah, they're there. They definitely live. They made their home there.
You know what you could do is just take some of that compost and then start adding, like turn it into a worm bin and start adding food scraps. Because
Oh
you
yeah.
see nematodes like this in an
Mhm.
environment, like a lot of people will be like, oh, there's 80 nematodes per drop. That's the best compost ever. But nematodes can only live like that when they have abundant food resource. So if
Yeah.
you take them and put them somewhere where they don't have those food scraps and like excellent excessive bacteria, you probably still won't get as many per drop just because of your recipe. But I bet if you take Brian's sample or even your compost and add more like food scrap to it, that you'll get like sometimes you'll see like 125 per drop. They're so
Yeah.
Yeah.
It's insane.
hmm
And this is actually a thing that we encourage quite heavily when we go to teach about composting is make connections with people that are local to you. So Raleigh lives an hour away from me, and trade compost. It's so important to be able to get diversity from other folks that are making compost, build a community in your area. So Raleigh, yeah, for sure,
Yeah.
down for making some trades.
We've got to get that
Um,
map up and going.
Yeah, for sure.
People
And
can
like,
yeah,
you
we
find
know,
do. We got to get the map.
closest.
I agree.
it was so cool comparing this to like, like Dan showing your compost. It was great because it was like seeing, I felt like the compost I made was like, it was like that same trajectory as yours. Like I maybe saw one nematode, but like, oh great, yeah, I'm seeing a good amount of testate amoeba, good seeing a good amount of fungi, which and like I know I got that backed up by Keish and Brian. Like, yeah, you're on the right track. Like even though I didn't make it like past that third turn, still after 6 months it aged into really nice compost. Gosh, look at this nematode party. This is
We
wild.
got a few questions here. Gay
Sure.
asked, do you use EM1 to add diversity?
I have not used EM1 at all. I have had clients that definitely use EM1 or Jam Korean natural farming, all those different type of more fermentation type of aspects. Um, yeah, you could. Didn't we have a student of ours, Keisha, that did add a lot of EM1 into their compost?
I do believe
I
that
thought we did.
Anthony uses EM1, but I might be wrong. Um, What's interesting, Matt Powers does he collects compost samples from everybody, and he got some of our Catalyst craft compost and then compared it to a lot of different, preps and um, anaerobic ferments and things like that, and did a nice chart. And what was interesting about our compost is it had all those organisms that you were trying to gain from the anaerobic ferments already in the compost.
In there,
I
yeah.
think we were kind of trying to show that earlier is as you're building a robust aerobic compost, you're also building a robust and diverse anaerobic compost, right? Because anaerobes and aerobes, they sit right next to each other in most cases, especially if the soil is healthy and there's good diversity. And so EM1 isn't something that you necessarily need to add for diversity because you're already creating it with your diverse inputs. These organisms exist in nature. So it's so lovely that we've learned how to bottle and bag and sell these products, but EM1 is a fancy name for a wild microorganism that's been on this planet doing good works forever, that someone was able to capture and then work out that it does well. So in a lot of cases, we don't need to add these anaerobic preps, but it's not going to hurt. I mean, I always say like, why
Right.
not add a little bit of it for diversity? There could be that there's organisms in there that aren't present in what you already have. So just wouldn't overload the system with it pure. I think with most of the anaerobic ferments, we like to dilute them like what 100 to 1, 1,000 to 1, sometimes even more. And I think they're fine to add, really. And then Kyle was asking about, do you worry about sodium levels with your crab shells?
I'm not really because I'm not using that much. I mean, I have big vermicompost bins. It's I'm shoot, it's probably about 40, 30 gallons per bin. And I'm adding a quarter cup every 3 weeks in there. So I haven't found any negative impact as far as that's concerned. And I use it, to help from a grit standpoint, to help the worms. And I'm sure, the breakdown of that is adding calcium. Those shells are high in calcium, adding that back into the mix as well. And managing a vermicompost bin, it takes a little bit of time to kind of get it balanced right. You got to the biggest thing I find with vermicompost is make sure you have enough carbon source in there. Woody material. I put in compost, aged wood chips go in there, paper bags go into it quite frequently. I really try to make sure I manage the carbon ratio. When you don't, then the bins tend to get a little I think facultative in that sense, and I don't get as good of a kind of the aerobic community that's happening there. Plus, also the fungi sources you get a little bit reduced. You can see through here, I've actually got quite a bit of fungi in this particular sample. Look at that tested amoeba.
Is there anything you
Isn't
don't
that
use,
pretty?
Brian?
I don't use cooked food, and I don't put a lot of meat into my bins either. Um, And I mean, I know people have used meat and you can use cooked foods too in there. I just tend to avoid them. So other than that, no, not really. I mean, it's any of my scraps left over. I pulled weeds from the garden. A lot of times, if I do that, I'll have root that will come along with it. So I'm taking some of those soil microorganisms and making sure that they get into the bin as well. Yeah, and it's just about diversity. I just really try to mix up the food stocks that are in there. Some vermicompost is made like an industrial scale where they only have a very limited amount of feedstock that they put into it. And typically you get a limited amount of microbial diversity when it comes to that. So for me, I'm just cognizant of, oh, I haven't added that to my worm bin yet. Let me go add that to it.
Diversity
And
and
overall, I mean, like I said, I'm happy with the way it's coming out.
That's one thing I really love about all of this is like you see this diversity in the microscope and you can save that in your mother pile to then keep increasing and keep adding nematode extracts. And then when you need to make massive large scale extracts, you have that diversity in your mother pile. And it's so cool. Like all of us here can create our own microbiology banks, our soil microbiology banks and then when we really want to scale up these application, like we have that. We can do really large scale extracts from even the stuff that we can make in our own backyard, which is so cool.
Yeah, unfortunately, Rich was asking about naked amoeba earlier, and I saw one earlier in here, but sometimes they'll go into an aggregate and you'll just never see them again. So
Mm.
unfortunately,
they
I
can be
haven't.
so cool. Check pond,
No.
like pond systems are a really good place
Oh
to
yeah.
find incredible naked amoeba. And when you're at the pond, try to think soil. So get plants, some organic material, any sort of mucky. And if it's too much sediment, sometimes it's hard to see, but man, just the right kind of muck on the side of a pond will have so many naked amoeba of all shapes and sizes.
I got a cool one here.
Oh,
Um,
what is that?
this is one of those ones that always kind of intrigued me. I saw them and I'm like, what is this ring? It's not a testing amoeba. It's not a test that's there. Anybody know? I have an idea what I think it is. Stomata. So when the stomata is the opening in plant leaves, this is where the oxygen carbon dioxide transfer actually happens. The material that makes up the stomata is really, really hard to break down. It's one of the last things to break down from the organic matter. And a lot of times you'll see these rings in there. So I'm pretty sure that's probably a stomata that it's there. All right. Well, any
Very
other
cool.
questions? We kind of getting to
How do you
the
manage
end.
worm bins in freezing conditions when not being able to get them inside to a non-freezing area?
Yeah. Um, Tough. I mean, is the worm bins freezing solid? If they are, then that's definitely a problem. If it's just got to where it's really cold and they've slowed down, then it's more of like a dormancy kind of thing. I would try everything I could to try to get them under cover and get them into an area where it doesn't freeze solid. If it freezes solid, that's probably going to pretty play havoc with the worms.
Mm. Mhm.
Although I personally, I don't have worm bins in freezing. It gets cold up here. These bins right now, it's been, you know, Fahrenheit wise, it's been in kind of the high 40s, low 50ish range. So it's cool, not really, really super cold. And the bins are thriving really well.
Hmm.
And they'll thrive really well when I push in summertime frame when it's more like 70 degrees outside.
Yeah, imagine them in the greenhouse too. They'll be eating all winter.
Yeah.
That's it. I had a question. So for folks who are interested, so the way that we're going to be teaching qualitative analysis, why don't we give people a small introduction to that for how we're um, doing that in the microscopy program. So Brian,
Sure.
Okay.
You go for it, Keisha, if you want me to, or you can do it.
Well, I guess we're gonna have two different formats. So we'll have one that's super, super simple. I call them quick checks, and it's more just talking about what you see very quickly. It's basically splitting up all of these different microorganisms into groups and giving yourself space on a paper to be able to write things out or type in notes. And I use this format when I'm at, say, I do a lot of events where I'll have, say, 300 people that I meet during the day, and 25 of them bring me samples and they want to know just something about their soil that's useful. And so I'll run these little 5-minute quick checks and at the end, you can look over and say, oh, wow, look, we're seeing, X amount of testate amoeba every few views. We're seeing fungi, but we're lacking nematodes. And it's just a really simple tool to be able to engage with your clients that might not want to pay a lot of money for a sample, but they want to have some sort of idea of what's living. You could liken it to what we're doing here. When Brian just threw that sample on, we were like, wow, that's great. Look at all these nematodes. Like, don't necessarily need to say that there's 60 per sample to say that there is an abundant amount of nematodes. He's doing
Right.
just fine on nematodes, lots of fungi. So that's one tool that we're going to have. And then we are still in the developmental stages of this tool. And if you know Compost Academy, you stay with Compost Academy. So I'm assuming this tool is going to change over time. This is kind of how we roll. We start out with an idea of something that we really want, like, oh, I think this will be helpful for the students. You guys use it, and then you come back with feedback and say, nah, I didn't like this, or I'd like to change that. And then we put work into making it better. So our first iteration of this tool is going to be coming out soon. It's in a spreadsheet, which means Brian's the one that put together all of the back end of the inner working. So do you want to talk about that, Brian?
Sure. Yeah. So, like Keisha was mentioning, we're kind of looking at a protocol to do qualitative analysis, and it really is more like a scorecard kind of thing. How much, not measuring. We're not going in and saying we saw a fungal hyphae of this diameter, this length, that kind of thing, and using kind of really advanced math or extrapolations to come up with pure quantities, right? That's a quantitative analysis. Our qualitative analysis is more, did you see fungal hyphae? Did you see fungal spores? Did you see protozoa? Did you see nematodes? And we look through a certain number of field of views. And if you did see so many of them, we kind of try to create a scorecard saying, it's in an area of microbial health of, yeah, probably good or meh or not so good, right? Um, and as Keisha mentioned, we've been working with clients for a long time frame. A lot of our clients do not need the quantitative. What they really want is just how has their soil evolved over time, and a qualitative analysis is perfectly fine for them. It saves them money, and it's also something that you can teach them to do. I've taught a number of our clients the here's how you use a microscope, and here's how you do your own qualitative analysis. And when something goes awry for them, like they're like, ooh, things have changed, either for the positive or for the negative, then it's a prompter for them to say, hey, let's have discussion around this. And they can show it on their camera, and we can look at it. And if they need to, we'll do a quantitative analysis to do the more precision kind of work. So, our philosophy, it really has evolved to a spot of saying we want more people to really understand how to do just good qualitative analysis so they get an idea of how much their soil is improving or regressing. And it doesn't have to be super precise like a quantitative.
Yeah, I think it's important to note too, it's like not just us, there's a lot of people working on this right now.
Yeah.
You guys are going to see a change very soon that the reality that the technology is bound up under one company is going to be gone. Everything's moving into open source now. And there's a lot of minds that are working on this. So yeah, just being frank, it's definitely going to change as we move forward. But we are in the process right now where we're exploring it, and it's very cool. It's like a soil score. But it's a little we've got some kinks to work out. We've got some things to
Sure.
play with, and it's going to be really cool to see multiple people use it and then give feedback so that we can really zone in on how we want this thing to function. But the
Yeah.
idea is everybody should be able to do it. We're trying to get, as simple as possible. Setting up a microscope, making samples, that stuff's hard enough. So, yeah.
Yeah, that's what we
Moving.
wanted to really demystify, right? Is the complexity around using a microscope. Once you get the basic skills of how to use a microscope, boy, the world is an oyster. You can go and start to observe all kinds of things, and it just helps. I mean, the more hours you put on the microscope, your eyes and your brain get trained to recognize certain patterns and shapes and things. And after looking through so many samples, it becomes very easy to make certain intuitive impressions about that soil, the quality of life. So what we want to do is just make sure that people have those skills to be able to use a microscope effectively, and they have the basic identification skills. And
Yeah.
then also build a community so that when you come across something weird, you go like, "What the hell is this?" There's a group of us that can go, "I think it's probably this. It's probably that." And we want to bring in do some more webinars with some, protologists, right? Nematologists.
I actually I think I had to Google it last time. I'm not gonna be as brave as you.
I almost said proctologist. I was like,
Know
wait,
those
no,
people.
that's
I do know those people. Their entire body of work is understanding protozoa.
right. Yeah, exactly.
Yeah, there's some
So
really lovely folks in the world that have dedicated their entire lives to that kind of work, and yeah, that's going to be exciting in the future. Learning more from them.
Yeah. Oh, we just had Jan sign up. Awesome. Excited to have you join us, Jan. You're going to join the microscopy team. Epic. Well, folks, I think we reached a good ending point here. We're almost at 2 hours. I'm going to post a link one more time for the folks who want to join us. Again, this link is just during this webinar where you get $100 off on the program. We really hope you join us because we're starting next week. You got a week to think about it, but we're rolling off next Thursday, and we got a really awesome program coming up. So just have that link again there on the bottom. Yeah, hop on to a microscopy program and join us. We can't wait to see your samples. We can't wait to see the progress that you can make in your soil and compost. And, like Dan, like Marie, become soil explorers, become people that can create micro banks all around the world and start improving soils no matter where you live. And again, thanks for joining us. It was a really fun session. Again, is great to have 55, I think we almost 60 people joined us. to explore microbes for 2 hours. It's always fun. It's always epic. And I'll get the replay out to everybody. I'll try to get that out today.
Hey,
Yay.
big thanks to Dan and Marie too.
Yeah, thank
Awesome for
you.
you guys to be able to come in and share your samples, and fantastic. We love it. This is what we want. We want more community, people to be able to share things. So this is exactly right up what we want to
And
And
have
taking
happen.
time
So
out of your
thanks.
life to make the compost so the
Yeah,
microbes
yeah,
exactly.
grow, taking time to check on them. You guys are epic. This is what we need. We're very
and,
pleased.
and
Thank you.
so
Thank
much experience and expertise you guys are sharing. That's so cool.
you.
Like you spotted nematode trapping fungi. Like we
Right,
got water
exactly.
bears. That's so great.
Yeah, love it.
All right, everybody. We'll have a great weekend and we'll touch
You
base next
too. Thank
week.
you very much.
Ciao.
Ciao.
Thank you all.
Bye.
Bye.